Dear students, welcome to my channel. The topic is Western blotting. Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment. Western blotting is a technique used to identify newly encoded protein from a mixture of proteins. The STS page is the prerequisite for Western blotting. When we examine the principle of Western blotting, immunochromatography, where proteins are separated by polyacrylamide gel electrophoresis according to their molecular weight. The principle is based on immunochromatography, where proteins are separated into polyacrylamide gel according to their molecular weight. Protein thus separated are then transferred or electron-transferred onto the nitrocellulose membrane and are detected using a specific primary antibody and secondary enzyme label antibody and substrate. Here it is given in short: protein separated by STS page, then transfer of protein to polymer sheet. Polymer sheet is exposed to radio label specific antibodies, then protein bands detected by specific antibodies are exposed to film. The procedure of Western blotting include preparation of tissue lysate, gel electrophoresis, transfer of separated protein out of the gel into a blot, then immunoblotting and detection. Here is the detailed procedure. First, preparation of sample lysate or tissue preparation. Take the sample, add ice cold PBS and lysis buffer such as RIPA buffer, which is commonly used buffer for maximum protein yield. The choice of lysis buffer largely depends on the localization of the protein of interest. Solubilization of membrane-bound protein requires stronger extraction detergent compared with isolated cytoplasmic proteins. Always use freshly prepared protease inhibitors, keep samples on ice and work quickly. Lysis buffer should contain protease inhibitors to prevent the degradation of the protein of interest. Cells are lysed by incubating on ice and later applying shear pressure using a pipette. The cell mixture is centrifuged and the pellet is discarded. The supernatant is a lysate which we will use for further processing. Western blots are typically performed under reduced and denatured conditions. These conditions will allow protein to be separated by their molecular weight rather than their native conformational shape or charge. To reduce and denature samples, dilute each in a loading buffer such as Lamley sample buffer. This buffer contains beta-mercaptoethanol or DTT to reduce disulfide bridges between cysteines, STS to assist in denaturing and to provide a net negative charge to the protein, glycerol to allow the sample to sink into each well, bromophenol blue to visualize the lysate and anionic buffer. Vortex each sample and incubate at 95 degrees Celsius for 5 minutes to completely denature the proteins. Now the sample is ready to load into an STS page gel. The next step is gel electrophoresis. Here, separate the individual protein in our sample lysate based on their molecular weight using a positive electrode to attract a negatively charged protein. To do this, we load our previously prepared protein sample into a commercially available polyacrylamide gel. Gels are available in fixed percentages or gradients of acrylamide. The higher the acrylamide percentage, the smaller the pore size of the gel matrix. Therefore, higher percentage of gels are better for low molecular weight proteins. Low percentage gel are useful for large protein, and gradient gels can be used for protein of all sizes due to their varying range in pore size. Prepare the gel by inserting it into the electrophoresis apparatus and filling it with a running buffer that is appropriate for gel chemistry. Rinse the wells of the gel with a running buffer and add a buffer to the chambers. Load the samples into the wells and load a pre-stained molecular weight ladder into one well. The ladder will allow you to monitor protein separation during electrophoresis and subsequently verify protein weight in your sample during later analysis. Close the electrophoresis unit and connect it to a power supply. Most units typically run 45 to 60 minutes at 200 volt or until the loading buffer reaches the bottom of the gel. During this time, the negatively charged protein in each sample will migrate toward the positively charged electrode, making their way through the polyacrylamide gel matrix. Next is the transfer. Transfer separated protein out of the gel into a solid membrane or blot. This is based on an electric field is charged to move the negative protein towards a positive electrode. Transfer can occur under wet or semi-dry condition. The steps of traditional wet transfer method are as follows. Start by removing the gel from its cassette and cutting the top portion containing the wells. Note the top left corner to indicate gel orientation. Float the gel in transfer buffer while preparing the transfer sandwich. To make the transfer sandwich, a cassette, sponges, filter paper, gel and PVDF or nitrocellulose membrane paper is needed. Notes: the top left corner of blotting paper to indicate blot orientation and incubate membranes in transfer buffer for 10 minutes. Create a stock by placing the following component from the black negative cathode to the red positive anode: sponge, filter paper, gel, membrane, filter paper and sponge. Use a clean roller with each layer to gently roll out any bubble that may be present, since bubbles will inhibit efficient protein transfer. Lock the cassette and place it in transfer apparatus containing a cold transfer buffer, ensuring that the cassette is properly positioned from negative to positive. In order to prevent heat buildup, it is beneficial to transfer with a cold pack in the apparatus or in cold room with a spinner bar placed at the bottom of the chamber. Close the chamber and connect to a power supply. Perform the transfer according to the manufacturer's instruction, which is normally 100 volt for a third to 120 minutes. Here the steps are given. First, load and separate protein samples on STS page. Then electrophoretically transfer fraction protein onto PVDF membrane. Block the membrane with neutral protein, that is BSA or milk casein. Then incubate the membrane with primary antibody specific to target protein. Incubate the membrane with HRP label secondary antibody specific to primary antibody. Then incubate the blot with chemiluminescent HRP substrate and expose to film. Here immunoblotting, after electrotransfer of protein into a membrane, we will now block the blot by applying a primary antibody specific for our protein of interest and then a secondary antibody that will recognize the primary antibody. So these are the steps given for immunoblotting. Start by removing the membrane from the cassette and rinsing in 3 times in water. As an optional step, we can verify the proteins were transferred successfully by staining the membrane with Poncio red. Incubate the membrane in Poncio for 5 minutes and wash with water until the bands are clear. After verification, the blot can then be de-stained by continuing to wash with water or TBS tween until the dye is completely removed. We need to block all area of the blot which do not already contain protein. This will prevent non-specific binding of antibody and reduce the overall background signal. Common blocking buffers include 5% non-fat dry milk or BSA in a TBS twin solution. However, do not use a milk solution when probing with phosphor-specific antibodies, and it can cause high background from its endogenous phosphoprotein casein. Incubate the membrane with blocking solution for 1 hour at room temperature under slight agitation. Decant the blocking solution and wash with TBS twin for 5 minutes. We are not ready to add our primary antibody. Dilute the primary antibody in a blocking buffer at the concentration recommended on the data sheet and incubate overnight at 4 degrees Celsius with gentle shaking. A recommend optional step is also use positive loading control antibody, which allows use to verify equal amount of total protein were loaded into each well and aids in troubleshooting by removing any uncertainties with the Western blot procedure. Next day, decant off the primary antibody and wash the membrane with a large volume of TBS tween and vigorous agitation 5 times for 5 minutes each. These stringent washes are extremely important for removing non-specific background signals. After washing the dilute secondary antibody in the blocking solution and incubate the membrane for 1 hour at room temperature at the concentration recommended on the data sheet. In our example, the secondary antibody is also conjugated to HRP for later detection. Decant membrane and wash the membrane with a larger volume of TBS twin with vigorous agitation 5 times for 5 minutes each. You are now ready for the detection phase. For detection, we start by mixing equal parts of ECL reagent into a 1:1 ratio according to manufacturer's instruction. We will incubate the membrane for 3 to 5 minutes without agitation. After incubation, decant ECL mixer and use a laboratory wipe to wipe off the excess solution from the corner of the membrane. Place the membrane in a clear plastic wrap such as sheet protector to prevent drying. We can now use a roller to push out any bubbles or any excess solution. Immediately develop the membrane. Both film and camera systems allow us to manually adjust the exposure time in order to ensure a picture perfect Western blot. Relative band densities can now be quantified with commercially available software. Proper molecular weight can also be verified by comparing band sizes to the molecular weight ladder. Next, we can see the uses of Western blotting. So, Western blotting is used to identify specific proteins or newly encoded proteins. It's a confirmatory test for HIV infection. Uses of Western blot is the most sensitive and specific test for determining the size and amount of protein present in any material. The confirmatory HIV test employs a Western blot to detect anti-HIV antibodies in a human serum sample. A Western blot is also used as the definitive test for Creutzfeldt-Jakob disease, Lyme disease, hepatitis B infection, HSV2, herpes type 2 infection. Here shows Western blot reactivity in one HIV-1 seroconverter. Dear students, thank you for watching. See you next class with another topic. Don't forget to leave a like and please subscribe my channel.